Yeah, so I'm Jess. I'm in the tail end of my PhD now in the Shepherd lab. And a main part, well, a huge part of my PhD has been to look at genomics and how we can think about how we can use genomics to inform or design autogenous vaccines to control Campylobacter in poultry. So I'm sure that the majority of people listening to this will already be quite familiar with Campylobacter, but I'll just recap. So Campylobacter is a really important pathogen to control because it's the leading cause of bacterial gastroenteritis in the world. It's very diverse, which makes it difficult to control. So there's lots of different lineages. It occupies multiple different sources, and it's found in the majority of mammalian and avian guts. But the majority of human infection comes from poultry, so eating undercooked, contaminated chicken. So by nature, Campylobacter is microaerophilic, so it doesn't like huge quantities of oxygen, so it can't survive in those conditions normally. But we find that when the chickens are colonized with Campylobacter on the farm, somehow strains are surviving and ending up on meat and causing infection in humans. So the Shepherd lab, before I started, they did some research and found that there's lots of different strains that are associated with different levels through the process. So there's an overrepresentation of certain clonal complexes, either found on the farm or found on carcasses or meat in the factory stage, and also separately found in human clinical cases. So that led to a study to do a genome-wide dissociation study to look at the differences between the isolates found at in these different groups to see if there was an association, a genetic association with the survival. So are there certain strains that have elements to them that allow them to survive through the poultry processing chain and end up in humans to cause clinical infection? And they did. So they found a list of genes that were associated with human clinical infection. And an important one of these is obviously aerobic survival. So that allows it to survive when it's broken out of the gut and ends up on the machinery and then contaminates meat. So there are lots of methods of control available that are implemented in farms and also in the actual processing factory. But due to the high demand of chicken, it's quite difficult to control the levels of Campylobacter. So vaccines are a well-known strategy to control infection, and they have been found to reduce levels of Campylobacter within chicken guts. So they can be effective against Campylobacter. However, due to the diversity of Campylobacter, it's difficult to find a vaccine that is cross-protective a lot against different infective strains. So we looked at different antigens that have been used previously in the literature that have elicited an immunogenic response in chickens. And we wanted to see just how variable these are within chicken or poultry-associated isolates. So we built a huge pangenome with over 1,000 chicken-associated genomes. And if you look to the graph on the left, the blue points are the antigens that we found in literature. And as you can see, these antigens that are common will have been previously used are the most variable out of all of the genes. And this indicates that, well, it gives an indication as to why it has been so difficult to make a vaccine for Campylobacter. So if we want a vaccine to work for Campylobacter, we need to target strains that are, well, we would like to target strains that are surviving, so the ones that are actually causing infection in the first place. We want to account for the high strain variability and generate cross protection, so we're targeting lots of different strains, and also it must elicit a long enough protection to keep the levels of Campylobacter low. And one good way that we thought of doing this would be to use autogenous vaccines. And autogenous vaccines are, they're very old-fashioned, simple, easy vaccines that have been commonly used in the farming world. And they're described as being, so they're an isolate or a pathogen that is taken from a particular herd or a particular farm. And that same isolate has been then used in a vaccine to inoculate the remaining animals within that herd or farm. And one benefit to using autogenous vaccines for Campylobacter is that you can use multiple strains within the same vaccine. So this is a diagram of the strategy that we had in mind. So this is kind of what we used to implement our vaccine. So chicks aren't colonized with Campylobacter in the first 2 weeks of their life, and we think that's because of the protection from maternal antibodies. And also broiler birds, because they live around 37 days, and this coincides with maximum colonization of Campylobacter, they don't live long enough themselves to elicit an immune response to a vaccine. So we thought that we would use this window as an opportunity to try our vaccines. So the idea was to select strains based off the genomic elements that are associated with survival that were found in the GWAS, and put these strains into a vaccine as they're most likely, well, they have a higher chance of survival. And then vaccinate breeder birds with these strains, and then we would expect the antibodies to be passed down to the eggs. And then as the maternal antibodies protect chicks from being colonized with Campylobacter, we're hoping that this gives an opportunity for non-survivor strains or strains that are less likely to survive to colonize the gut before the survivor strains do. So one of the natures of autogenous vaccines is that they have to be isolated from the same farm and used on the same farm. So we worked with a poultry company in the UK, and we sampled across 5 different broiler sheds to understand the strain variation that's circulating at that time, and also to select isolates to include in a vaccine. And we sampled the sea key, because that's where we would expect the highest concentration of Campylobacter within the bird. And also we sampled from the neck skin, but we kept the neck skin packaged and refrigerated to replicate how this would be done in real life and retail when it goes into supermarkets, because we thought that this would maximize our chances of finding strains that are likely to survive poultry processing. So we, at the end of the sampling stage, we found 135 different strains of Campylobacter. So we sequenced these, and so the tree on the left, so I compared them to lots of different common chicken-associated clonal complexes, and we found 4 distinct clonal complexes. So the 3 of them, so the red, green, and yellow, are common chicken-associated clonal complexes that we would expect to find on the farms. And these have all been found in human clinical cases as well. But the purple this belonged to a selection of isolates that we didn't know the chemical complex of. So once we'd sequenced them, I looked to find, well, we then wanted to select 4 isolates, 1 from each kernel complex to include in the vaccine, but we also wanted them to have the maximum survivability. So I looked within all of the isolates and selected 1 isolate from each kernel complex that had the most survival-associated genetic elements. And the plot on the right, so the red is if the genetic element is present, and then white is if it's absent. So we made the vaccine. It was grown up. So we used the 4 different strains, and they were grown up into huge quantities using fermenters. And then the antigen was inactivated with formalin. So these were whole cell, and they were suspended in an oil-based emulsion. And then we vaccinated the birds. So we vaccinated them at 2 doses, 14 and 18 weeks of age, and we vaccinated them at the breeders at the breeder rearing stage. That's at the very beginning of the poultry distribution system. And then we had a corresponding control non-vaccinated breeder farm, and we made sure to follow the same flocks throughout the chain to avoid any mixing with non-vaccinated flocks. So how do we know if the vaccine has had an effect? So for one, we would like to see a reduction of surviving strains on the neck skin in the vaccinated cohorts, the vaccinated broilers. And also we would like to see an alteration in the gut strain population structure compared to pre-sampling. And then we also would like to know if there'd been an immune response in the first place in the birds. So we want to look for protective vaccine-specific antibody. So we sampled, so we gave the breeders 2 doses, and then when they reached, or when we expected them to reach around peak immunity age in the vaccinated, we then sampled the progeny of the breeders at this point to look for Campylobacter. So we sampled around 900 isolates from 5 farms of vaccinated progeny and also 5 farms of control progeny. And then we sequenced these. So at the moment, I've only got around to 2 out of 5 farms sequenced, but the isolates that we found post-vaccination are totally different from the strains that we found pre-vaccination. So the kernel complexes in the pre-vac were all common chicken-associated kernel complexes, and the isolates that we found so far in the post-vac there's ST21, which is more of a host generalist kernel complex, and also we only found C. jejuni in the pre-vaccination sampling, but this time we found C. coli as well, and also we found ST353, which is more like a commensal associated kernel complex, but we still have the remaining 3 farms to see the rest of the variation. And then we also looked at colony forming units. We did an enumeration of all of the isolates. So the top, I've got to label it, the top 2 box plots, these are from pre, and then the bottom 2 there from post. So the difference between the CQ load was pretty much the same pre-vac and post-vac, but we weren't expecting to see a reduction in the CQ. We were But the more interesting thing that we wanted to see was that we wanted to see a reduction of the Campylobacter numbers surviving on the neck skin in the vaccinated cohort. There's little difference between the control neck skin post-vac and the neck skin pre-vac, but we do see a slight reduction in the Campylobacter surviving on the neck skin in the vaccinated cohort. So another way of seeing if there had been, to see if the vaccine had been effective, was to see if there was an immune response in the birds that we were vaccinating compared with control cohorts. So along the process, along the vaccination process, we took samples of blood from breeders, from vaccinated breeders and control breeders, and we took eggs at the same time that we took blood. And then while we sampled the progeny for the Campylobacter, we also took blood from straight after hatch, around 2 weeks of age, when the maternal antibody would be expected to decrease, and then just before slaughter, which is around 37 days of age. So so we did ELISAs with the blood and the egg samples, and we looked to see in the breeder blood, we would expect to see a much higher concentration of antibody, vaccine-specific antibody in the vaccinated breeders compared with control. So red are the vaccinated the vaccinated breeder blood, and the black are from the control cohort, and each bar represents one of the 4 strains included in the vaccine. So the breeder blood is a really good result. So we can see clearly that there's much more antibody found within the vaccinated cohort rather than the control cohort. So that's a result that we expected and wanted. And then in the egg yolk, we wanted to see much more vaccine-specific antibody within the egg yolk of the vaccinated cohort, which we do at time point 1, and then it slowly starts to decrease a little bit, but then starts to increase again at time point 3. But you do start to see some cross-reactivity in the control populations as well. And then we checked the broiler blood. So we still see vaccine-specific antibody in so straight after hatch, which is what we would like to see. And then it decreases at time point 2, which is also what we would expect to see as maternal protection decreases. But then you do see an increase at time point 3, and we expect this is probably cross-reactivity from natural infection that's happening on the farms. So is there evidence that the vaccine is having some effect? And we think that there is. We think that it's having some effect on the population structure, but there's still work left to do. So we still need to sequence the remaining farms. There's 3 farms left over to see the variation of clonal complexes within those farms and see if they match the ones that we already have or if these are totally different. And also it'd be interesting to model the effects of the vaccine. So as the vaccine is whole cell, we don't know which antigen is causing an immunogenic response. So we're going to be using machine learning to characterize the differences between pre and post isolates, and then use the machine learning to model the effects of the vaccine. So, for example, how many poultry isolates could be knocked out by the 4 strains that we have included, based on known antigenic variation, but this is still ongoing work. Okay, so that's the end of the talk. I'd just like to say thank you to the lab that I've been doing my PhD in, so the Shepherd Lab, and also I'd like to thank Jose and Yori from Oxford University, who will be doing the modelling with, and also Jay, who's in Roberto Laragioni's group from Surrey, who have very kindly carried out the ELISAs. So thank you.